In this video, we talk about the prokaryotic cell. Here, already we discussed what is meant by prokaryotic cell. Prokaryotic cell, it's nothing but the absence of the membrane-bound nucleus. So, here the prokaryotic cells which are here the bacteria cell, blue-green algae, mycoplasma and PPLO. These are the prokaryotic organisms. You see here we have to discuss the structure of the prokaryotic cell. Here the structure of the prokaryotic cell, here the cell envelope of the bacteria which contains three layers. Here the cell envelope of a bacteria which contains three layers. One is a glycocalyx. Second one is a cell wall. And third one is a plasma lemma or plasma membrane. Yes, see here it is a typical bacterial cell structure, and see the first one is a outer layer, that is a capsule, yes, first one is a glycocalyx. Here it is a glycocalyx. Here glycocalyx in some bacteria which is in a thick condition, and it is referred as capsule. Here it is a glycocalyx. Here it is the outer layer. It is a thick layer, and it is a very tough condition which is present in the bacteria. Here it is the outer layer of the bacterial cell wall. Here it is a tough condition in some bacteria, that is called a capsule. But in some bacteria these outer layer, this glycocalyx which is arranged in a loose sheath. Here it is that is called a slim layer. But in some bacteria it is arranged in the tough condition, that is called capsule. And second layer that is a cell wall. See here it is the second layer. Glycocalyx or capsule, it is the outer layer. Here it is the outer to second layer, that is a cell wall. Second outer layer, that is a cell wall. Here it gives a shape. It gives the perfect structure and shape of the bacteria, and it prevents the bacteria from the bursting condition. Yes, next third layer, outer to inner, that is the third layer, that is plasma lemma or plasma membrane. Here it is the innermost layer. Here it is the innermost layer. Here it is the semi-permeable membrane. Like in eukaryotics. Here it is the semi-permeable. Here it is a, if it is necessary to the any other substances, it gives a semi-permeable, which means here it gives the permission to the outside and outer to inner to outer. It gives the permission, so it is called a semi-permeable membrane. That is called a plasma lemma. It gives the semi-permeable condition. To the molecules. And here it gives a lot of extensions. These extensions are, see here, these extensions are protrude into the cytoplasm. These are called mesosomes. See here. These are the mesosomes, the extensions of the plasma membrane. Here the extensions of the plasma membrane, they are forming the vesicle-like structure, tubule-like structure, that are called mesosomes. Here, what are the main function of the mesosomes here? The mesosomes which are a major role in respiration and secretion process. They are used in the secretion process, and it is also forming the cell wall formation. Okay, and DNA replication also, it is also help in the DNA replication process. And here And here the outermost layer, it is also having some appendages like pilus, Here it is some projections. Here some projections. These are called pilus. These are called pili. Here pili. The more number of these pilus structures are called pili. In some bacteria, fimbria are also there. In Fimbria also there. Pili and Fimbriae. Pili and Fimbriae. Pili and Fimbriae. Pili and Fimbriae. Those structures are also present on the outer membrane of the bacteria. Here the pili or pilus, which are present in the gram-negative bacteria, which are present on the gram-negative bacteria, which is used in the conjugation process, which is attached to the other bacterium, and it is the horizontal gene transfer mechanism. It is also used in the horizontal gene transfer mechanism. That is the role of the pilus. And the Fimbriae, here the Fimbriae which is present in the gram-positive bacteria as well as gram-negative bacteria. Here the main function of the Fimbriae are it is used to attach the substrate. It is used to attach the substrate or host. Substrate or host. Here it is a bristle-like structure. And here within the bacteria, see here in the bacteria semi-fluid structure, it is called a cytoplasm. Here it is a cytoplasm. And in that cytoplasm, here the thread-like structure is there. Thread-like structure. Here it is a naked DNA. Here it is called a nucleoid. Here it is a nucleoid or DNA. Here it is a naked condition. There is no. Here the nucleoid cannot enveloped by any membrane. So here it is not. It's not enveloped by any membrane. So here it is a naked DNA. And addition to this DNA, the small circular DNA is also presented there. This is called a plasmid. This is called a plasmid, small circular DNA, which is also plays a major role in the gene transfer mechanism. Yes, here the based on the consumption, based on the staining procedure, the bacteria which is classified into two categories, that is gram-positive bacteria and gram-negative bacteria, which was proposed by the Christian Gram. And these are two classes. What are the two classes? It was proposed based on the staining procedure. Gram-positive bacteria, gram-negative bacteria. Based on the staining procedure, which means here the gram stain which is absorbed by the bacteria, that is called gram-positive bacteria, and those who are not absorbed the staining, that is called gram-negative bacteria. Here the gram-negative, the pilus which are present in the gram-negative bacteria, Fimbriae which are present in both gram-positive and gram-negative bacteria. And see in this structure, see in the cytoplasm, the ribosomes are also there. See here, observe this. The ribosomes are also there. Here, which means here it is not bounded by any membrane. Which means here the membrane bound organelles are all absent. In the prokaryotic organisms. Here only the ribosomes are only present in the prokaryotic organisms. Because of ribosomes are not having any membrane. They are not having any membrane. So the ribosomes are present in the prokaryotic organisms. So here they are having the more number of the ribosomes are having on a single mRNA. Here it is the mRNA, messenger RNA, which is composed of many number of ribosomes. Here these are the ribosomes. These are called polysome. Polysome means many. Polysome means many number of ribosomes are arranged on a single mRNA. Many number of ribosomes are arranged on a single mRNA. That is called polysome. Which is here the main role of the ribosomes are, it gives the protein synthesis. It plays a major role in the protein synthesis. Protein synthesis. Here these the ribosomes are, what type of the ribosomes are present in the prokaryotic organisms? Here the 70S type of the ribosomes are present in the prokaryotic cells. Here these 70S ribosomes are made up of 50S plus 30S subunits. These are made up of 50S plus 30S subunits. And in the cytoplasm, here another non-membrane bound organelles. Here the inclusion bodies are also there. Inclusion bodies. Here the inclusion bodies are non-membrane bound organelles. These are the reserve food materials. These inclusion bodies are reserve food materials. Here the phosphate granules, the synophysin granules, the glycogen granules, these are the non-membrane bound bodies which are present in the cytoplasm of the prokaryotic cell. So this is about the prokaryotic cell structure. I hope you understood. Thank you.